Welcome back to EQBloom. Today we're exploring one of the most fundamental machines in all of biology, the ribosome. This organelle is so central to life that every living organism on Earth, bacteria, plants, animals, humans, use ribosomes to make the proteins that keep them alive. Ribosomes are the translators of genetic code. They take a message written in RNA and convert it into a chain of amino acids. A protein that can fold into enzymes, receptors, antibodies, hormones, muscle fibers, and every structure that makes us who we are. So let's walk through how ribosomes work, why their structure matters, and how they decode mRNA with stunning accuracy, and also why translation is one of the most essential processes in all of life. To understand how ribosomes function, we need to dissect their structure. Every ribosome has two subunits. In eukaryotes, these are the 40S small subunit and the 60S large subunit. In prokaryotes, the 30S and 50S subunits. Each subunit is made of two components, ribosomal RNA and ribosomal proteins. But what surprises a lot of students is this. The ribosome is mostly RNA, not protein. And even more importantly, RNA performs a catalytic reaction. That means the enzyme responsible for forming peptide bonds, the heart of protein synthesis, is not a protein enzyme. It's a RNA acting as a ribosome. This is a clue to the ancient origins of translation. It supports the idea that early life on Earth may have existed in an RNA world where RNA stored information and performed enzymatic functions. So in the ribosome, proteins play a supportive role, stabilizing the structure, but the RNA does the critical chemistry. Now that we know what the ribosome is made of, let's explore what it reads. mRNA is the instruction tape, a long linear sequence of nucleotides. Ribosomes read the sequence 3 bases at a time, forming codons. Each codon corresponds to a specific amino acid. The process always begins with a start codon, almost always AUG. This codon codes for the amino acid methionine, but more importantly, it acts as a flag that tells the ribosome to start reading here. From there, the ribosome steadily moves along the strand from 5 to 3. When it reaches a stop codon, translation ends. These codons do not code for any amino acid. Instead, they recruit release factors that trigger the ribosome to release the completed protein. The elegance of this system is remarkable. The ribosome knows exactly where to start and where to stop because the mRNA carries these built-in signals. If mRNA carries the blueprint, then tRNA is the decoding tool that translates that blueprint into amino acids. tRNA molecules have a distinctive structure, often drawn like a clover leaf with two critical regions. One, the anticodon, which matches specific codons on the mRNA. Two, the amino acid attachment site, which holds the corresponding amino acid. Each tRNA is paired with one amino acid, and that pairing is enforced by a special enzyme called an amino-acyl tRNA synthetase. This enzyme ensures accuracy. It's like a molecular quality control officer. If a tRNA is loaded with the wrong amino acid, the entire protein would be built incorrectly. But synthetase enzymes proofread and edit mistakes, giving translation an extremely low error rate. tRNA essentially acts as the translator between two languages, the nucleotide language of mRNA and the amino acid language of proteins. Now let's bring everything together step by step. Initiation. Translation begins when the small ribosomal subunit binds to the mRNA. A special tRNA carrying methionine finds the AUG start codon. Once this match is made, the large ribosomal subunit clamps into place and forms a complete ribosome. Elongation. This is the engine of protein synthesis. New charged tRNAs stream into the ribosomes, A site. If their anticodon matches the codon, they're allowed to enter. Then the ribosome performs its catalytic action. It forms a peptide bond between the new amino acid and the growing chain. The chain is always held in the P site. Then the ribosome shifts forward, a movement called translocation, bringing a new codon into position. This happens incredibly fast, 10 to 20 amino acids per second in eukaryotic cells, even faster in bacteria. Then termination. When the ribosome encounters a stop codon, no tRNA can bind. Instead, a release factor enters the A site, triggering the ribosome to release the completed protein. The ribosome then falls apart into its two subunits ready to begin again. One of the biggest misconceptions is that free ribosomes and bound ribosomes are different. They're not. They're the same ribosome performing the same chemistry, just in different locations. Free ribosomes float in the cytosol. They make proteins that will stay inside the cell, such as cytoskeletal proteins, metabolic enzymes, nuclear proteins, and mitochondrial proteins. Bound ribosomes attach to the rough ER. They make proteins destined for secretion outside the cell, the plasma membrane, lysosomes, and the endomembrane system. What determines a ribosome's location isn't the ribosome. It's a signal peptide at the beginning of the protein. This short amino acid sequence tells the ribosome, send this protein to the ER. So the ribosome moves based on the message it's reading, another example of how instructions are built directly into the sequence. As we wrap up, let's zoom out. Ribosomes matter because every protein in your body, every enzyme, every antibody, every receptor, every hormone originates here. Why this matters? All proteins come from ribosomes. Everything that gives cells structure and function depends on translation. Translation error causes disease. Mutations in RNA, ribosomal proteins, or tRNA synthetases can lead to neurological disorders, developmental problems, and cancer. Viruses hijack ribosomes. They don't bring their own, they use ours to manufacture viral proteins at high speed. And lastly, translation is the fastest process in cell biology. A single cell can make thousands of proteins per minute. Ribosomes are the engines of life, constantly reading, decoding, and building molecules that make living systems possible. And thank you for learning with EQBloom.